Thanks, Julianne, and also to the whole team for inviting me. And I was flipping through all the lists of past speakers and the topics. I was really impressed and thought that, wow, this is a venue where really the latest technologies in AI and genomics are kind of being revealed in real time, which is really exciting to see. And so, yeah, a lot of my time, especially between sort of 2019 and 2023, a lot of my time was focused on running Deep Genomics as the CEO. But in 2023, we got a new CEO so that I could focus on research and leading the research teams and getting into the weeds, learning about what a transformer is and things like that. And so I've really enjoyed that. So it feels good to be back focused on research. And one of the things that Deep Genomics that we've been doing too is really trying to start to open source more of what we do and put our models out there and make them publicly available. So I'll talk a little bit about that as we get going. So the talk, what I wanted to focus on today is that if I look at the list of previous speakers in terms of topics, a lot of in the last couple of years, a lot on foundation models for what I'll call genome biology, so for DNA, RNA, and protein. And the question is, can we use those foundation models to properly analyze genetics data to identify targets? Can we use those foundation models to design genetic medicines, right? And so really my talk today is about crossing that chasm, because we'll often see people publish foundation models and maybe they'll do a little bit of benchmarking. But from the point of view of someone who's been working on medicine now for the last 10 years and, in deep genomics, a lot of those benchmarks aren't really indicative of impact in medicine. And so, deep genomics is definitely at the intersection of AI and medicine. And so we have a lot of insight into what really works, what doesn't work. And so I'll talk about foundation models. I'll talk about the gap between foundation models and our ability to use them in medicine and in particular drug discovery. And really kind of end on kind of a comment, I would say, more about the situation and this chasm, if you like. Sounds pretty dramatic, but anyway, between the two and sort of what we need to solve in order to move forward. Okay, so very briefly, at Deep Genomics, our machine learning team, we work directly on drug discovery, and so we build foundation models, but the people who build those foundation models are actually looking at patient data, developing oligonucleotides or guide RNA therapeutics for therapeutics. So first of all, I just want to make that point that at Deep Genomics is really a close connection between the work of our ML team and computational biologists, and as you know, everyone who surrounds those people. And drug discovery. Our focus now is building AI with partners because we feel that's the best way we can have the largest impact in industry. We'll have our wholly owned drug programs in the near term. And then in terms of the longer view of the company, we really are looking to revolutionize drug discovery. That's why I got into this as an opportunity to do that. And so it's not just about Deep Genomics, it's about the whole community. It's about biotech, it's about big pharma, the whole thing. So here's a picture of the team. I'm going to be talking about a lot of different contributions that these team members have made, and I won't have time to properly recognize each and every one of them. An incredible team, and I just feel very privileged to be working with these exceptional people to do this work. Now, Deep Genomics, in terms of what these people do, they're first of all very multidisciplinary. We don't have like computer scientists and biologists. They can speak to one another's language. Huge amount of humility and respect for one another, which is key. But roughly speaking, you can think of Deep Genomics as having machine learning. We've got about 7 people there right now. Actually, it's probably more like 10. Engineering, and that includes like machine learning engineering, genetics and targets. So those are people who can do genome-wide association studies or look at do KOL interviews to understand how a genetic mutation in the context of a disease might be addressable. We have lab automation, drug discovery team, and other as well. Yeah, so if you're interested in this kind of endeavor, I'd love to hear from you, and you can apply. We've got a link there that you could go to look into how you could join our team. Okay, so today I'll break it down into 3 parts. First of all is causal foundation models of genome biology, which is dear to a lot of people's hearts, I'm sure, on this call. I'm going to show you some results, and these are results Two types of results. The first type of result is kind of more like a benchmarking result. The second type of result is showing you the application of the foundation model to drug discovery. And so that's kind of the bridge I want to talk about here. And then the third one is really kind of what the gap is and what's going on. And I would really say I capture it this way that, our foundation models speak the language of RNA or protein or DNA, whereas drug developers and LLMs speak English or another human natural language. And those are two completely different languages, of course. The language of genomics is in nucleotides, chemical interactions, things like that, not English or any other human language. And so I think that's really the interesting bridge that we need to cross is kind of a language bridge. More on that later. All right, so just a bit of context. First of all, my experience in the field over the last 10 years, I would say that first of all, if you just look at industry standard, you would identify a variant associated with the disease, a mechanism of the biology, a mechanism of your therapeutic molecule, and so on, through to safety and tolerability, and so on. Now, traditionally, you would de-risk each of these step by step. So you'd have a hypothesis, you would go and test it in the lab, you get a yes/no answer, and then you'd move on. That was sort of the approach. So very little look ahead, very little synergy between these tasks, and very little prediction, right? And so instead of testing thousands of hypotheses, you test one or two. What Deep Genomics and other AI companies started doing about 10 years ago is building narrow AI systems. So you'd have an AI system that was good at adding information to analyzing patient mutations. Another AI system for looking at variant biology, like splicing or something. And all of these different AI systems would address different problems. And at Deep Genomics, up until about 3 years ago, we used to proudly talk about having 40 different machine learning models to address all of these different problems to help us do everything from target discovery all the way through to safety and tolerability. Now, we proudly talked about that. In retrospect, the whole system was not scalable. Imagine trying to maintain 40 different models. Imagine getting new data. How do you properly re-engineer that data? 40 different models. How do you retrain 40 different models? How do you validate and test those models in the context of medicine, where you're going to have to use orthogonal assays, ask questions about what this means for patients? These are difficult questions to answer. How do you do that for 40 different models? And how do you deploy those 40 different models? And then also for some tasks, the models overlap. Like you might have a model that looks at a patient mutation and predicts splicing, another model that looks at a patient mutation and predicts how that'll change polyadenylation. Well, how do you combine the outputs of two separate models? Because they won't be calibrated in terms of their risk probabilities and uncertainties and so on. Nonetheless, that's what we did, and other companies did this as well. By the way, a lot of other AI companies just focused on one narrow slice. Like, for example, Atomwise was a company that was really focused on protein as a small molecule, not focused on target identification, not focused on safety and tolerability. And so many of these other AI companies actually just focused on one of these slices of this overall pie. Now, what's been transformational for us at Deep Genomics and other companies as well, and obviously the academic community is hot on the trail of this, is foundation models, where we can build a foundation model that really can understand and model all of these different tasks all at once. And it's trained on huge amounts of data. It achieves synergies between these tasks when training. And so, for example, transcription and splicing and polyadenylation are all interrelated in terms of how the action works. So accounting for that interrelationship during training is important. It's one model, so when we want to increase the amount of data, maybe 10 times more data, we can just retrain that one model. A huge training effort, but it's done once. If we want to increase the architecture, again, it's one model, increase the size of the architecture. Also, as I'm sure a lot of you already know, for a foundation model, it's often good at tasks that it had not been designed to solve initially. And so our foundation models at Deep Genomics, like BigRNA, will be useful for a task that we hadn't even thought about applying it to when we built the model. And so that's really important. Kind of, you can think about that as like zero-shot learning, or you can just think about it as fine-tuning or whatever. But the point is, the model can be applied to new tasks really easily. Now, deep genomics, foundation models, there's a lot of foundation models. Deep genomics is at the intersection of two major fields, RNA therapeutics and causal foundation models. And it's kind of exciting, I think, for all of us that, just in the last couple of years, there have been a series of Nobel Prizes really in these areas, both in AI and machine learning, but then also in RNA. So both mRNA vaccines and then last year, Nobel Prize in microRNA. So a lot of action here. We think that's an exciting place to be. Now, one comment that's kind of fun that I wanted to make is attention is not all you need. You need models that respect the causal rules of biology. So this, of course, is a bit of a tongue-in-cheek comment of, attention is all you need. But it's not sufficient to have, for example, a transformer architecture, which can be very powerful. You also need to be talking about what are you predicting, right? And so you can go at a problem where you're trying to predict correlations, or you can attack a problem where you're trying to predict causal relationships. In both cases, you could use a transformer. Now, in biology, deep genomics has always been really focused on causality. All of our machine learning systems predict causal relationships that can be tested in the lab and validated. And of course, causality is important if you want to create an intervention like a genetic medicine, or understand how a patient mutation leads to disease. But here's an interesting result from Gonzalo and his collaborators. If we look at classifying complex trait variants, and I won't go into the details of these results, but just a snapshot. If you look at classifying complex trait variants, and you look at models that are not causal, they're trained like hyena DNA, for example, or Evo2. These are models trained on DNA sequence. They're not trained to predict function. And compare those to models like Informer and Borzoi, which are trained to predict function. CAD is sort of a system that has many different models built into it, and the new version of CAD does predict function. What's noticeable here is that the models that are mapping causal relationships or predicting function perform a lot better in terms of AUPRC for classifying those complex trait variants. And so, I think, and a lot of people think that predicting causality is important, and this is just a fun example to show that. Now, at deep genomics, how do we think about this? So one of the wonderful things about genome biology is that we know a lot about kind of the block diagram or the flow chart, if you like, of how different things depend on one another. You cannot say that for pathophysiology, or you cannot say that for cell signaling, or more complex types of biology, but you can say that for genome biology. We know about transcription, we know about splicing and polydenylation. We know about microRNA targeting and mRNA stability. We know about translation. Now, I'm not saying this is perfect. There are many components that we're not aware of, or we're missing, or we have very incomplete information about, but at least we do have a flow chart of, the central dogma, if you want to call it that kind of shows us what's going on. And so what's important at deep genomics is we look at this block diagram and we collect data sets, either internally or public data sets or through CROs. We collect data sets that are basically capturing the inputs and the outputs of these causal relationships. For example, DNA sequences and pre-mRNA sequences, recognizing that the pre-mRNA is generated from the DNA. So if we collect data on both of those, we can then build a machine learning model that captures that relationship. And so this diagram here just shows you all the different kinds of data sets that we're looking at, and others in the community have looked at, to build foundation models that capture causal relationships. I'll just add that, there's different, for each of these processes, different things you can imagine doing. You can collect observational data. You can collect data from different tissue types or different disease states. You can do perturbation experiments where you intervene somehow in the gene. It could be CRISPR, or you could perturb it with an oligo. There's different ways you can perturb it, and then look at the consequences. So each of these each of these little orange rectangles is just showing you the kind of data you could collect, but I'm saying there's different ways you could generate that data, from observational through to perturbation. Now, I wanted to comment, this comes to this chasm thing. The impact in medicine, from my point of view, requires domain knowledge of causal biology and also this collaborative multidisciplinary team that I was talking about before. And it just means that, you might have somebody who's good at building a foundation model, but they need to make sure that in the end it can actually be used for drug discovery. And so this little diagram here, it's sort of like collaborative multidisciplinary teams on the x-axis, so less collaboration, less multidisciplinary in the far left, more on the far right. And then in terms of the vertical axis, domain knowledge of causal biology, a lot of domain knowledge on the top and very little in the bottom. And from my point of view, you're going to get high impact if you're in the upper right quadrant there. So these are models built by multidisciplinary teams and with expertise in terms of how they would be deployed in practice to create value. All right, before I start talking about the technical, sort of the more technical part of the talk and the foundation models, I wanted to give you a sense of how the team at Deep Genomics thinks about the problems we solve, and that's what this rather complicated diagram is meant to get across. So in terms of foundation models, we'll build foundation models. We'll also ingest them like publicly available models. We'll also fine tune them, make them better and faster and application specific. And so I'll talk about examples of all three of that when we get into the details. But I wanted to talk about, what kind of biology are we modeling? And you probably already gathered this from the previous slide. But we look at DNA and RNA biology. So that's DNA and RNA sequencing modeling, protein biology, and then also systems biology. So you can think about single cell data sets and that kind of thing. Now, I'd say systems biology is the one where there are still a lot of uncertainties. And if you ask, what's the real deal versus just correlation, I would say that for RNA and DNA, we've got the real deal, a lot of causal relationships that we predict. Systems biology is still a little correlative and still not, we still haven't really nailed that as a community, I would say. But we still do work in that area. Lots of different data sets, from proprietary data sets, public data sets, genome, single cell, perturbation data. The thing I want to emphasize is at the bottom here, the lab in the loop idea. So we have our foundational model platform. It's capable of delivering novel insights. We spend a lot of time on validation and optimization and making sure it's scalable. But really critical is this lab in the loop idea. So at Deep Genomics, we've got about 25 people in our wet lab. And they're part of the closed loop of model improvement all the time. And we have cycles that can be as short as a couple weeks or other cycles that take a few months. But when we build our foundation model, we'll then validate it and test it using different kinds of experimental data, not just the same data that was used for training, but maybe we train the model on observational data, but then we're going to test it on some perturbation data or something like that. It also generates data for retraining, of course, and fine-tuning the models. So it could be large amounts of data, which I'll talk about an example of that, a large amount of data that we use to train a foundation model, or it could be a small amount of data that we use to fine-tune the model and make it better for a particular application. The applications we do look at are shown on the far right. So therapeutic design, target prioritization, discovering novel targets, translation and safety, and then also patient stratification. And that's really not a comprehensive list of applications. When partners come to us and say, hey, could you work on this? Quite often it might not exactly fit within this list, but the answer is yes. Okay, so let's look at some data now and actually see what's going on. So here's a picture that I just showed you before of the central dogma, and there's really three buckets I'm going to look at. The first one is RNA processing. The second one is mRNA stability, and then the third one is DeepADAR for RNA editing. And they're sort of in order from most general to most narrow. So DeepADAR is really about designing RNA editing molecules. So for the first bucket, BigRNA is a model we published in 2023 that I'll talk about a little bit. And then Flash RNA is a model we just put out there about a week or two ago. It's publicly available. You can download it and apply it. Put it on the website. And both of these models, as I said, are for RNA processing. And I'll tell you about both of them as we go through the talk here. So BigRNA was trained on about 1 trillion signals in total and predicts a diverse set of regulatory mechanisms. And when I talk about 1 trillion signals, I'm talking about basically DNA nucleotides, RNA-seq measurements per nucleotide. I'm talking about different proteins, different microRNAs, different tissue types. So there's many different dimensions which you can think about the data. So it's all listed on the far left there, the types of data that were used. The training and validation, I won't talk about that in detail. I'm gonna focus on just what the model does, because I kind of think if I get that across, then you can imagine in your head how you might train the model, given the inputs and the outputs for training. And I will say that this did involve a multidisciplinary effort. And so if you look at the paper, there's quite a large number of authors, and that's because we tested the model on 12 different drug discovery problems, ranging from target identification all the way through to developing therapeutics. And there have been some really good recent publications, the Alpha Genome publication recently, which is great to see, but not much there in terms of actual drug discovery in medicine. And so we really wanted to focus on the drug discovery problems when we were looking at BigRNA. Now, in the far right, what you're seeing is again the central dogma, and everywhere you see like a Deep Genomics logo there is a process that we can use BigRNA to make a prediction for. And so not only is there a lot of complexity in terms of how you use it for drug discovery, but there's also a lot of complexity in terms of all the different processes that you could make a prediction for using BigRNA. All right, so let's look at the model. Yeah, and I'm not going to show you a big complicated diagram or a bunch of math or anything like that. I'm actually just going to give you the intuition for what the input is and what the output is. And then, as a machine learning person, you can imagine if you have a whole bunch of examples of the input and output, you can train it to minimize root mean squared error or whatever you'd like to minimize. Okay, so it's a seq-to-seq model. The input would be a gene up to 1 million nucleotides. Now that could be the DNA sequence or it could be a pre-mRNA sequence. Either one could work, and up to 1 million nucleotides. So that goes into BigRNA. It's got a bunch of stuff in it. And then the output of BigRNA is essentially a predicted set of tracks and roughly 3,000 different tracks corresponding to different molecular phenotypes. And so examples of a track would be a prediction for what you'd see if you did RNA-seq in cortex. A prediction of what you'd see if you did RNA-seq in liver. So that's shown in red, for example. Another track would be riboseq, so it can predict what you'd see if you were to perform riboseq for that gene. Protein binding events, again, it'll predict where you'd expect each and every one of 669 RBPs to bind. Similarly, microRNA binding, which is shown in purple there, and then RNA methylation. There are other tracks as well, but these are some that I wanted to highlight. Now, you might kind of be thinking, wait a minute, okay, you have the data for these tracks, so what's the usefulness of the model? Well, once you've trained the model, it's capable of inductive reasoning. And so once you've trained this model to predict tracks from the gene sequence for different species, different tissues, and so on, you can now look at a patient mutation. So I can just computationally change a G to a C or whatever in the DNA, run it through BigRNA, and then we'll see how all of these different tracks change. And so, for example, we might see that an exon is skipped. And we might also see that a particular RNA binding protein ceases to bind. And so now you kind of have both a prediction of the impact of the patient mutation on molecular biology, namely exon skipping, and also a bit of understanding of what might be going on. So maybe the patient mutation disrupts the protein binding site, and when the protein doesn't bind, that causes exon skipping. And so it's a lot of information, 3,000 different tracks, each one of the same length as the gene. It's a lot of information, but we have systems that can analyze those tracks and then come up with a mechanistic hypothesis of what's happening. And then we've validated that in our wet lab numerous times. So that's just one application, kind of you can imagine, whatever you'd like to do, you can do it. You can simulate the effect of an oligonucleotide just by blocking the nucleotides where at the input where it would bind and then running it through the model. You can think about DNA or RNA editing. And so given that patient mutation, you can now try introducing a whole bunch of other edits and see if the other edits recover the exon. And so if there's an edit that does recover the exon, now you could conceive of maybe having an RNA editing therapeutic to achieve that edit. Or if you're interested in the mRNA design, you can look at the 3' UTR for that gene. And you can ask, if I change nucleotides in the 3' UTR, do I see increased expression? And if I see increased expression, then BigRNA is making a prediction that I could modify the 3' UTR and get a more stable mRNA sequence. So that could be a good approach to designing an mRNA therapeutic. So yeah, kind of anything you might imagine doing, you can then sort of do. I'm not saying it's perfectly accurate. Of course, it's not. For some things, it's quite good. For other things, it's not as good. But you know, I think a big part of this is capturing the imagination of the person who might want to use this for drug discovery, and then and then talking to people like that and feeding back their input to make the models better where they need to be better, creating new heads, new outputs, new kinds of predictions that will help them make drugs. So that's kind of the idea here. So I'll pause here and just any questions kind of about the approach we're taking and about what I've just talked about here for BigRNA.
Probably clear for this audience, I guess, no? Yeah, this audience is quite familiar with
Good. Okay, good. Good. I can get on to the next stuff here. Okay, so I won't spend a lot of time on the simple benchmarking. Because as you know, benchmarking is quite complicated. There's different ways you can benchmark AUC, AUPRC, correlation, and all of this. I just wanted to point out that when we compared BigRNA to other techniques on a variety of different tasks, here there's 5 different tasks that are in the paper that you can look at if you want more detail. It performed well across the board compared to other techniques. Again, I'm not going to go into the details here because it would take a lot of time. All right, so now I want to talk briefly about flash RNA. So flash RNA we just put out there a couple weeks ago, and so the BigRNA model we did not make public. Borzoi is a similar model that is public, and so what we've done here is focus kind of on the Borzoi side of things, and then use flash attention to improve the self-attention efficiency. So that's what flash RNA is. And we are planning to release a version of BigRNA near the end of the year that'll sort of be everything that BigRNA is about, also with flash attention. But I guess the thing to notice here is that if we compare it to Borzoi, we're seeing about a 6-fold increase in speed for inference and about a 2.5-fold increase for training. And so our hope here, and part of what I'm interested in here, is trying to make these models as available as possible to the academic community, who might not have as much GPU compute power as companies, right? And so we're going to be doing more of this, I'll say. So flash RNA is just our first step, but we're going to be doing more of this kind of thing to try to help other people use these kinds of models and train them, and then of course innovate, come up with new ideas with smaller compute budgets, which I think is pretty important for the community. When we benchmarked flash RNA, so this is a student at the University of Toronto who did this work on eQTL and RNA half-life data. These are the results we obtained. So what you're seeing here on the lower left is eQTL effect size, Spearman correlation, Spearman correlation for the predictions. And you're seeing alpha genome. These are the single models. So alpha genome, Borzoi, and flash RNA single. And so pretty similar in terms of effect size. And the differences, I would say, are not that meaningful in terms of impact on medicine, right? Correlation of 0.406 or 0.396 when you take into account other factors that are crucial for actual downstream application doesn't really make a difference. Anyway, the point here is they're pretty similar. When we look at ensembles, we look at flash RNA and Borzoi, we get 0.43 for Borzoi, 0.45 for flash RNA. And then when we look at the distilled models, flash RNA is getting 0.45, alpha genome's getting 0.48. So all this is to say pretty similar. Like if you look at these 3 categories of the single models, the ensembles, and the distilled models, we're seeing pretty comparable performance, but at much higher computational efficiency. We also looked at predicting half-life, and we compared flash RNA to Saluki and ribo-NN. And then here you're seeing the Pearson correlation for RNA half-life. And in these cases, flash RNA performs better. 0.81 versus 0.62 and 0.77, 0.73 versus 0.66, 0.71. Again, I'm not sure how much these differences will make in terms of actual medicine, but nonetheless, as I said, sort of benchmarking is something we're all interested in, so we wanted to show you these numbers. Okay, so now let me talk more about the drug discovery side of things. So that's kind of the benchmarking side. What about actual drug discovery? And that's, I think, kind of where we all need as a community to do more work to make these models as useful as possible. So this example here is to show you the BigRNA can discover causal disease variants and also corrective oligos. And so the model input, which we're showing, is a one-hot encoding in the upper left, and obviously that's just a small snippet of the entire sequence. Can be fed into BigRNA, and then it'll make a prediction for all of these different tracks. And I'm showing you the RNA-seq track for liver, and focused around exon 6 of ATP7B. And so BigRNA predicts that you will see inclusion of this exon 6 ATP7B. When we include a mutated sequence, so this is a patient mutation in the context of Wilson disease, BigRNA predicts that that exon will be skipped, and so that's shown in the
There's
sort of the
something
orange part.
there.
What was that?
She was just going to bung it on the sandwich. I was going to make carbonara, but she didn't want it.
Sorry, I didn't understand. You're going to say what?
I think it was a mistake.
Okay,
She
all right.
muted herself.
Okay. All right, so BigRNA predicts that this mutation will cause skipping of the exon. And what we can now do is just synthesize the effect of an oligo, basically set all of the inputs to 0.25, right? So ACGT, we just set them to 0.25, where the oligonucleotide would bind to the RNA sequence. And then we can run that through the model and make a prediction. We did this with a whole bunch of oligonucleotides, so basically tiling the oligo along the length of the mRNA. I'm just showing you one of them here. And when we run that through BigRNA, it predicts that the exon will be included. And that's what you're seeing here in green. And so that way we can now think about using BigRNA to analyze the patient mutation and how it will impact molecular biology, and then also design a therapeutic that could restore the original function. We also went and tested these in the lab. So we CRISPR'd the mutation into a cell line and saw that indeed that mutation does cause exon skipping. And prior to this work, the community thought that the mutation, which is an exonic mutation, led to a missense amino acid, which led to a non-functional protein. And we showed that that's not the case. It's causing exon skipping. And then in the lower right, you can see that the predicted effects of these oligos, and now it's not just one oligo, we're looking at all of the oligos tiled across that exon, matches up quite well with the measured effects. So the predicted is shown in gray, measured is shown in orange, and the correlation is 0.91. I mentioned before the importance of sort of application-specific tuning. And so the community comes up with foundation models. How do we use them in medicine? A big part of this is fine-tuning those models for a particular disease context. And so we wanted to show an example of that. And so here, what we did is we looked at ALS, and we looked at, in the context of ALS, how is molecular biology changed? And one thing that happens in the context of ALS is TDP-43, which is an RNA binding protein, is sequestered in a condensate, so it's not available to participate in regulation of RNA processing. And so what we did is we downloaded TDP-43 knockdown data that was in eye neurons, and then we fine-tuned BigRNA using that data. And that gave us what we'll call BigRNA ALS. So it's just a model that's been fine-tuned with TDP-43 knocked down. And then we used that model to score patient mutations. So we looked at a set of 157 known patient mutations for ALS. And for each of those mutations, we ran it through BigRNA, and we ran it through BigRNA ALS. And we scored each mutation in terms of changes in splicing. Basically, we just looked at the splicing track and came up with an aggregate score for whether that mutation would cause a change in splicing. And then we ranked them, and then we looked at those ranked mutations. So what you're seeing in the left rectangle here under BigRNA is the 157 mutations that are ranked. And then we looked at a positive control, which is a Curalis-Lilly ALS program, which is a particular variant, as you can see there, RS129-73192. And it's ranked 33 in this list. When we look at BigRNA ALS, it goes up to rank 4. So this positive control, if you like, pops up to be higher in the list when we use the ALS fine-tuned version of BigRNA. And then you might be asking, what about the other, what about rank 1, 2, 3? So those are novel. They have not been previously looked at. So we're studying those. And then there's others like 5, 6, 7, 8 in that list that are also interesting. So this is how we can use the model for an application-specific use in this context for scoring patient mutations. You could also imagine application-specific oligo discovery or DNA editing or RNA editing discovery as well. For example, an RNA editing or a DNA editing therapy may not work if TDP43 is not present. And so you want to know that before you go ahead and do all the work of synthesizing these guide RNAs and testing them. All right, so the next thing I'm going to talk about is REPRESS. So this is a model for mRNA stability. And mRNA stability is important, of course, in the context of mRNA therapeutics, where your goal is to design an mRNA that will be stable and then transcribed. But it's also relevant for a lot of other applications. Patient mutations can cause a decrease in mRNA stability. So classifying a patient mutation as pathogenic and understanding its mechanism, mRNA stability can be important there, and also designing other kinds of genetic medicines. This one's also available. The code for REPRESS is available online as well. So in this case, we generated a huge amount of data internally in collaboration with the CRO, and then used that data to train the model. Here I am showing the architecture, but I won't go through a lot of the details of the architecture. But again, what's the input? What's the output? Here, the input would be the 3' UTR. We capture a little bit of the exon as well, just to give a bit of context. But you can see that in the diagram. So in this case, it's a 12.5 kb context. And then the outputs, there's two types of outputs here. Again, they're both tracks. One of them is microRNA AGO2 binding as measured with an assay. And then the other one is degradome-seq, which is where you look at the degraded end of the 3' UTR, and then you get a read on the end. And so what you're seeing here is a profile of where the degraded end of the 3' UTR is. So those are two different types of data that we use to train the model. Again, on the benchmarking side of things, first benchmarking, then therapeutics. On the benchmarking side of things, I won't go into all the details here, but we looked at 7 different models for microRNA binding, target scan all the way through to miTAR, mirrorbind, dMeso, other models. And then we also looked at 7 different validation tasks. So for each of these, if we look at REPRESS, for example, we made sure that, of course, it was not cheating and trained on the data that it was then tested on. In most of these cases, we're testing it on data that's really orthogonal to the data that was used for training. And so there's no way that we could have cheated. And we try to ensure that for the other methods as well. Wherever you see a red spot, that means that model performs the best among all the other models for that task. And then dark blue means it's performing the worst out of all the other models. A white rectangle is a situation where we're not showing you the result because oh, that's interesting. We're not showing you the result because it's the data set is the same type of data that was used for training. And so even though we were careful to hold data out for testing, if it's the same type of data, even that can create a bias, right? Same sort of experimental setup, same cell line or whatever. So we're just not showing you those because those would all kind of be red. So you can see that REPRESS generally performs quite well compared to these other techniques across all these different tasks. Now, for those of you who are like machine learning gurus, we also looked at different kinds of architectures. So we looked at both the microRNA binding and also the degradome performance. And then we looked at REPRESS, the REPRESS architecture, which is, by the way, about 33 million parameters. We looked at a dilated CNN, Mamba, transformers, and also a vanilla CNN. And just to show that this model architecture that we came up with for REPRESS does outperform the other model architectures. All right, so let's move on to some applications for drug discovery. So in this case, we're using REPRESS to design antisense oligonucleotide siRNA and mRNA therapeutics. So 3 little vignettes. I'll show you one example of each. So first of all, antisense oligonucleotides. So we looked at UTRN ASOs, a publication on those from Sengupta et al. In 2020. And there's 2,037 different oligos. We basically just scored those oligos using REPRESS. Again, how much the oligo is predicted to increase mRNA stability. And then we compared them to what was found in that paper experimentally. And we're showing you an example here. The little blue the little blue bump here with a little orange rectangle beneath it is a prediction from REPRESS in terms of the microRNA binding events. What you see down below is now a prediction without the ASO, again, using REPRESS, and the prediction with the ASO. So just simulating the effect of the oligonucleotide, again, by setting the inputs to sort of 0.25. And you can see that the oligo is predicted to, if you look at the track without the ASO, prediction with the ASO, you can see that the track is predicted to, you can see that the oligo is predicted to decrease microRNA binding there. And that, by the way, also does correspond to the lead microRNA blocking ASO identified by Sengupta. So there's a positive control there. If we look at siRNAs, slightly different diagram here to kind of look at what's going on. Here we looked at 1,000 different siRNAs from the Houston data set. Again, we scored each siRNA using REPRESS. And I guess the key here is that we're looking at siRNAs that have a seed match, but not perfect complementarity, right? And so these are siRNAs that'll have a greater or lesser effect. They're not going to all have a really, really strong effect. And then we analyze them again in a similar fashion. In this case, to produce a plot of the fraction of good hits identified versus the number of siRNAs tested. So basically, how many siRNAs would you need to test in order to achieve a certain hit rate, which means kind of out of the known true hits that have an effect, what fraction of those are you achieving? So if you look, for example, at a 50% hit rate, using the Deep Genomics model, this REPRESS model, you'd use roughly 5 times fewer. You need to screen 5 times fewer oligos to achieve that 50% hit rate compared to the industry standard technique. The last example I'll show you is for mRNA. And so here we're looking at 3' UTRs. And what we did is, as I was talking to you about earlier on, we just looked at all sorts of different ways you could mutate the 3' UTR. And then for each of those, we looked at the measured mRNA level, log2. That was data internally generated in our lab. And you can see a pretty good correlation between the two. That is, when the machine learning model REPRESS predicts a high score, you're likely to have a lower mRNA level. And so we're improving this model currently. It's a good sign that we're getting this correlation. We want to achieve higher correlation. But currently this is the result. Okay, the last, I do want to wrap up in the next 3 minutes, so there's a bit of time for questions here. The last one I'll talk about is DeepADAR, which is for RNA editing. So natural RNA editing is basically a process that converts A to Gs in your RNA. It's happening all the time in your RNA right now. Basically, a secondary structure is formed in your RNA, and then this ADAR machinery recognizes an A with a complementary secondary structure and then edits the A to a G. And we can basically co-opt that machinery by using a guide RNA that targets a particular patch of RNA and simulates that secondary structure so that the ADAR machinery recognizes it and then edits an A to a G. So now you can think about this in terms of therapeutic design. If you have a particular nucleotide that was a G to A, you can now switch it back to A to G. That would be one approach. That's just correcting it or fixing it. You can think of a coding region, and you can think about non-coding. So in the non-coding region, it could be a regulatory element. So change an A to a G, and maybe you create a better binding site for a protein, which can lead to expression increase. In the case of the coding region shown on the far right there, that's where you might change an A to a G in in an exon change the amino acid and lead and create a different protein. I won't go into as much detail on DeepADAR for the sake of time. I'll just quickly show you our pipeline here. We've trained on 1 million naturally occurring edit sites. That leads to DeepADAR. We can then use it for predicting natural editing, or we can use it to predict guide RNA induced editing. And it achieves a Spearman correlation of about 0.76, and that's a zero-shot prediction in terms of editing efficiency. So quite good. And when we fine-tune it using additional data, we get even higher correlation. What we're doing right now with DeepADAR is using it to design RNA therapeutics. We hope to have 2 to 3 programs in mice in the first quarter. And what I wanted to emphasize here is this tight feedback loop. We design the guide RNAs using DeepADAR, we manufacture and screen them in a cell-based assay, sequence and analyze the results, and then retrain DeepADAR. And that's on about a 2-month cycle, so quite quickly. Last comment here before I wrap up is we are seeing good scaling laws, which is exciting. When we look at, say, REPRESS, plot the amount of data on the x-axis and how well the model is working, it's getting better and better as we add data. So as you might guess, we're generating more data. Similarly, if you look at model size, so for example, number of parameters on the x-axis and performance on the y-axis, we're also seeing improvements as we increase the size of the model. So those are curves that are very promising in terms of what we'll do next. All right, my final remarks have to do with this barrier, this language barrier, RNA to English. And no results here, really. I just wanted to make a couple comments. I mean, first of all, there's a lot of excitement about LLMs, and maybe we don't need these foundation models. The people on this call here work a lot on different types of machine learning and foundation models and so on for biology. Maybe we don't need them. Maybe we can just use ChatGPT, right? So that's an interesting question. So on the left, I'm kind of showing you the schematic that makes me think that's not the case. There's kind of all of biology. There's genome biology. And then within genome biology, there are biological foundation models like BigRNA or Borzoi, Evo2, SCGPT, all these different foundation models. And my prediction would be that those models understand things that the current LLMs out there just don't understand. Mainly because, of course, the LLMs are limited to what humans have written. So we did an experiment and actually found that this is the case. What we did is we looked at patient mutations where the mechanism of the mutation had been published. So by the way, we're kind of letting the LLMs cheat because these are mutations where the mechanism has been validated experimentally and published. We then looked at each of those mutations with just an LLM. We put it in the LLM and said, what's going on here? And then measured the accuracy of classifying the mechanism of the patient mutation. And then we also gave the LLM the BigRNA output. And so with that additional information, asked it to classify the mutation. And you can see that for both Gemma 3 and Llama 3.1, and actually this is true for other LLMs, we see a significant improvement in the ability to classify those mutations when you give it the foundation model information. And this is just the first cut at the experiment, right? Our first attempt. So why am I showing you this? It's because LLMs do know how to talk to drug discoverers. Like LLMs can speak English. They can interact with the researcher. Our foundation models cannot directly interact with the researcher. So this result is kind of an interesting combination of the two, right? Putting the LLM together with a foundation model. The other comments I wanted to make is that as a community, we're generating more and more of these foundation models. There's currently over 50 foundation models. And this is a plot of number of foundation models, cumulative number of foundation models by year since 2018. And you can see it's growing rapidly. So the other problem that our community has to deal with is even if you have a foundation model and you put a lot of work into making it available to drug discovery or drug hunters, what happens like 2 months later when another foundation model comes out, right? So how do we close that gap? And so, my final slide here is really just an idea, which is that we should be thinking about building agentic AI that uses foundation models to collaborate with drug developers at scale. So think about a networked agentic AI, it could be based on ChatGPT, could be based, your favorite LLM, doesn't really matter. But the point is that the agentic AI knows how to use all these foundation models, knows how to formulate inputs to those foundation models, knows how to interpret the outputs of those foundation models, and then can provide a very simple, rapid, and effective interface to the researchers who are developing genetic medicines or trying to find targets, that kind of thing. And those could be people in companies, could also be people at universities, researchers, or biobanks, what have you. But that's what I just wanted to leave you all with, is kind of that idea of agentic AI that can connect the dots there and basically translate the language of foundation models to English. All right, I'll stop there. Thank you.